Welcome back to Biology 102. Today we're going to start Chapter 13 and we're going to discuss fungi. Now here looking at the scientist, this is Heinrich Antoine de Berry. He's a German scientist born in 1831 and died in 1888. Is considered the foremost expert on fungi, cyanobacteria, and plants in the mid-1800s. At a young age, he was introduced to botany through his physician father, who encouraged his participation in natural history excursions. So he had a parent who would constantly take him outside and constantly show him different things that were around his environment and tried to get him into the scientific field. He attended German universities in Heidelberg, Marburg, and Berlin, earning his medical degree in 1853. His investigations into the natural history of plants and fungi as a hobby, and it's going to lead to his first publication and botany professorship at the University of Tübingen, Germany, in 1853. He's going to teach botany at the German universities, including Freiburg from 1855 to 1867, Halle from 1867 to 1872, and Strasbourg from 1872 to 1888, where he's going to continue to examine the life history and morphology of fungi. Scientific accomplishments related to fungi are going to include proposals of the first phylogenetic classification systems, discovery of their parasitic and pathogenic abilities, detailing their complicated life cycle, and describing their mutualistic association with algae to form lichens containing the term symbiosis. And he does this in 1879. And one of the first things we're going to talk about with this is going to be the evolutionary history of fungi. Fungi is going to evolve from a marine-dwelling, flagellated, unicellular protist about 1.5 billion years ago. With this, the oldest fungal fossils are going to be discovered in marine and estuarine environments. That's going to suggest that early fungal species were like extant members of Chytridiomycota. The closest living relative to fungi are going to be marine protozoans known as Nuclearia. Fungal groups today are going to lack flagellas. Remember, those are going to be the extensions that come off at the back of the cell, the front of the cell, or they could be found on both sides, but they're used to make them move. They're used in a whip-like motion. So this structure was believed to have been lost either as a solitary event or over the course of several independent evolutionary events. Now, terrestrial fungi are going to diverge from aquatic flagellated ancestors about 550 million years ago. First, they're going to appear in the fossil records during these Silurian and Denovian periods, with most fungal groups appearing by the late Carboniferous. Recent fossil discoveries are believed to point to earlier origins of terrestrial fungi during the Ediacaran period from 635 to 632 million years ago. Loss of flagella and evolutionary hyphae are going to advance their terrestrialization of fungi. So as they start to lose these things, we're going to see them start to move across the ground and through different environments. Fungal proliferation and diversification have been attributed to ecosystem destabilization and the collapse following the extinction of photosynthetic vegetation during the end of the Permian and the Cretaceous mass extinction events. Now, with this classification, we're going to look at different scientists. Of course, we always go back to Carlos Linnaeus in 1735, first classified fungi as flowerless plants. And he looked at these at cryptogamia. Then we're going to have Ernest Haeckel in 1866. He proposed a three-kingdom system, but fungi is going to remain part of the plant kingdom. So up until a certain point, they're actually going to think that these are part of the plants, or the plant kingdom. Herbert Copeland in 1938 is going to remove fungi from the plant kingdom, and he's going to place them within the kingdom Protista upon proposing his four-kingdom classification system. So now we're starting to separate them out, and they do realize that they don't belong in with the plants. Robert Whittaker from 1959 and 1969 is going to place fungi into their own kingdom and make a significant modification to the kingdoms by proposing four major divisions. So current fungal classifications that we're going to go over. Okay, although their combined use of molecular and morphological data has greatly improved the current fungal classification system, it's not provided a comprehensive phylogenetic classification system that's going to be accepted by mycologists. Now, when you talk about myco, you're talking about fungi, and of course, phylogenists, these are going to be specialists. So these are people who specialize in funguses. So first comprehensive classification system was going to be proposed in 2007. It's going to recognize that there's going to be 7 divisions, while more recent 2019 systems are going to recognize that there's going to be at least 9 major divisions of fungus. Most mycologists are going to recognize fungi as a monophyletic kingdom in the domain Eukarya. It consists of 5 major divisions. So remember, Eukarya is going to be the division or the domain that consists of any organism that is going to have organelles, membrane-bound, nucleus, and things like that. So with this, like we said, it could be unicellular, it could be multicellular, but it's going to fall in eukaryotic domain. It's going to store carbohydrates as glycogen, like animals do, and so that's its form of starch. As a plant, it's going to store it as starch. There's sessile. Sessile means that it stays in one place and it does not move. So they're unable to move except for the flagellated chytrids. They do have cell walls, which provide structure and protection, and they're going to be composed of some certain things. Now, these cell walls were once what was thought to have put them into the plant groups, but we've gone in and we realize now that they do not belong in with plants. So with these cell walls, they're going to consist of a substance called chitin. That's going to be a complex structural polysaccharide. We have glucan. That's a polysaccharide making up over half of the overall structure. And we're going to have some glycoproteins. These are going to be unique associations of proteins and carbohydrates that maintain cellular shape. They help to provide protection, and they increase the chitin synthesis, or making or production of chitin. Some of the features that we're going to see with fungi is that they usually have a thallus. This is a vegetative body of the fungus. They have mycelium. This is going to be a mass of interwoven filaments of hyphae. And of course the hyphae, which are going to be the long filamentous branches of fungal cells. You could have 2 types of hyphae. You could have what's called coincidic. That's going to consist of single, elongated, multinucleated cells. Or you could have septate. That's going to be divided into multiples or individual cellular compartments. So hyphae are going to help to facilitate the absorption and movement of nutrients into the body of the fungus. Mushrooms are probably what you think of most when we talk about fungi. They're going to be a temporary reproductive structure, or lots of times we refer to it as a fruiting body, and it's going to be made up of an aggregation of these hyphae. Now, fungi are going to reproduce both asexually and sexually by billions and billions of spores, which are going to be typically dispersed by either the wind, water, or animals. And once they're dispersed, they're then going to grow into new hyphae. So with the reproduction, asexual reproduction could have 4 different reproductive modes. It could go through mitosis, which is going to be your spore-bearing hyphae that produce genetically identical haploid spores. You could have budding, where a bud of a parent cell is going to split and produce a daughter cell. You could have fission, and that's going to be a division of parent cells into 2 daughter cells. And you could have fragmentation. That's where a portion of the mycelium is going to separate and grow independent of the colony. With sexual reproduction, you're going to have 2 different types. The 2 main stages are going to be plasmogamy, where 2 compatible hyphae, you can have a plus and a minus, are going to fuse together to form a dikaryotic hyphae. As dikaryotic hyphae become tightly packed together, they're going to form that fruiting body structure that you notice and see growing usually above ground, which is the mushroom. You also have what's called charogamy. That's where 2 haploid nuclei are going to fuse to produce a diploid zygote that undergoes meiosis, and it's going to help to produce haploid spores that are going to be released and then dispersed. Saprotrophy is going to be another thing that we need to talk about as far as with nutrition. So you could have heterotrophs, that are going to acquire their nutrition by consuming other organisms, or you could have the saprotrophy. That's going to be a heterotrophic mode of feeding fungi that they employ, which involves digesting dead or decaying organic matter. It's going to be a two-step process. First, they have to digest. So that involves using the hydrolytic enzymes that they're going to release to break down complex organic molecules. Once they release these and it starts to break down that dead decaying matter, they're then going to absorb it. So small molecules are absorbed through the hyphae and transported through the mycelium. Other fungal species may acquire nutrition through parasitism or forming a symbiotic relationship with other organisms. Roughly, there's going to be about 148,000 described species of fungi on Earth. This represents only 10% of several million that we believe actually exist out there. There's going to be between, we think, 1,500 and 2,500 new fungal species that we discover each year when we look at the different numbers that come in. This is accelerated over the past several years with the inclusion of molecular data to fungal studies, because now they can go in and think and actually check to see if things are related or not, or if they're completely new species. So things that we used to think were belonging together in the same group, now we know due to DNA that they do not. So new discoveries within the kingdom are going to be lagging due to their cryptic lifestyles. Lack of morphological characteristics, we have convergent evolution, and we even have a limited number of experts that actually can study this and go in and actually figure out which ones are going to be new species and finding these. 5 primary divisions are going to be based on morphological and reproductive characteristics. We have Clitridiomycota, Zygomycota, Glomeromycota, Oscomycota, and Basidiomycota. So those are going to be our 5 primary divisions. With the Clitridiomycota, they're going to represent the simplest and oldest fungal groups that consist of species characterized by the presence of flagellated reproductive spores called zoospores. These are going to be found in aquatic and terrestrial habitats. Most are going to have chitinous cell walls, although some cell walls are going to be composed of both chitin and cellulose. And they're going to produce spherical fruiting thalli bodies that are going to consist of filamentous rhizoids, which are going to help to anchor the fungus and help absorb nutrients. With the Zygomycota, they're going to consist of terrestrial species such as Rhizopus stolonifer, that's going to be your black mold, that can reproduce both asexually and sexually to produce reproductive spores called zygospores. And you can see that in the picture below. Glomeromycota are going to be a distinct group of terrestrial fungi that are going to form a mutualistic relationship with plants to produce a specialized type of mycorrhizae known as arbuscular mycorrhizae. And so you can see those here. Ascomycota are going to be the largest division of terrestrial species in the kingdom fungi and representatives of the subkingdom Dicaria. They produce dikaryotic hyphae, asexual spores called conidia, and ascospores, which are going to reproduce or which are produced sexually. And this is going to include things that you might know as food products like yeast and truffles. With the Basidiomycota, these are going to be terrestrial species that are going to be composing subkingdoms of Dicaria. They produce dikaryotic hyphae as well as distinctive reproductive structures such as mushrooms and spores called basidiospores. Now with each of these you can go back and you can look at the different life cycles that each one goes through. Some of the ecological benefits of fungi are going to be that they are recyclers. So due to their saprotropic behavior, it's going to help to facilitate and release and return those trapped nutrients and dead decaying organic matter back into the ecosystem so it can be transferred and used throughout other organisms. Some symbiotic relationships that they would have are going to be things like fungal epiphytes. These are organisms that are found on the surface of plant structures which help to provide them with shelter and nutrients. You can also have fungal endophytes. These are going to be found within the tissues of plant structures and receive shelter and energy from the plant while providing protection to the plant from harmful soil organisms. They may have given rise to an extant mycorrhizae. Remember, extant means that things are still around. Extinct means that they are not. All right, so with the mycorrhizae, they're going to exist as a mutualistic relationship between fungi and plant roots. These are believed to have been instrumental in the emergence and success of plants on land, and it's supported by the fossil records and molecular data. Mutualistic relationships are going to involve fungi receiving carbohydrates that are produced by plants and fungi helping that plant increase the surface area and permeability of their roots. We're going to have two types of mycorrhizae that are going to exist to help maintain plant health and productivity. We have endomycorrhizae and ectomycorrhizae. With the endomycorrhizae, this is going to help to penetrate root cells through those cell walls and forms what's called an imbricule. This is an invagination. That's where it bends in on itself. That's going to increase the surface area for absorption of water and nutrients. It's also going to provide protection and induces plant growth by stimulating the production of plant hormones. Ectomycorrhizae are going to exist as hyphal sheets that surround the root tips. These are going to help to increase their absorptive surface area. So it's going to help to provide protection, and it helps the plants tolerate inhospitable soils. So some of these symbiotic relationships that different funguses have are going to be things like their symbiotic relationship with a lichen. This is a group of slow-growing organisms that form a symbiotic association between fungi and cyanobacteria, or between fungi and green algae. The fungus is going to receive carbohydrates from the photosynthetic symbiont, and the cyanobacteria, or the green algae, receive protection and nutrients from the fungus. Scientists believe this relationship may be more parasitic than mutualistic, with some fungal species receiving more than 90% of the photosynthetic byproducts. So they're kind of taking that from the cyanobacteria, and they're taking it from the other green algae that could be working with it. Another looking at this with the lichens, we have three main types. We're going to have what's called the crustose that you see here on the tree bark that grows on rocks and trees. You have fructose, which is going to be a shrub-like form. You can see that here in the middle picture. And you have foliose, which is going to be a leaf-like form. These are considered ecological contributors because they help to improve the quality of soil for plants, and they help to monitor air pollution. So if you see that these start to die out, or you have an area that doesn't have a lot of these, it could be a possible area that has air pollution. So some of these are going to have some benefits to humans. They help us produce alcohol, so humans have been using yeast for centuries to produce alcoholic beverages without knowing that it was a fungus, or knowing its important role in alcohol fermentation until the mid-19th century. And that was thanks to Theodore Schwann and Louis Pasteur. With human nutrition, we're going to use different types of funguses, including morels, portobellos, shiitakes, and highly prized truffles. And these are going to be used in our food sources for different types of nutrition. So they're highly nutritious, and lots of people seek these. With the production of food, we can use different types of yeast and different types of funguses to help us to produce breads and cheeses, including gorgonzola and brie, which are characterized by their appearance and distinct flavors. We also use them with the development of antibiotics. So it's important that we use these because we get antibiotics that are derived from these fungi and develop them and introduce them into society. So this is what happened when we came across penicillin. It was actually a fungus that started to grow on a petri dish that kept the bacteria from growing around it. Some other things, we have cephalosporin and cyclosporin. They're going to help to positively impact human health as well, along with the penicillin. So that's going to end us on our unit on fungi. I'll see you back here with our next unit.